feel honored to introduce Dr. Mohapatra, who hardly needs any introduction. But still, as I have been asked to say or talk about his brief CV for the benefit of the youth, I'll take a few minutes for that. Dr. Mohapatra is a blend of scientist and administrator. I have seen him when he was a student. I have seen him when he was an active researcher. I have seen him in management positions, in the present position which he is holding as my distant successor. He has very important positions in the Indian Council of Agriculture Research. More important was his stint as a director of Central Rice Research Institute and director of IRI, and then as secretary there and DGICR. And he has a lot of publications, a lot of awards, and a lot of role played nationally as president of the National Academy of Sciences, and having contributed in the international level as vice chair of governing board of CRISET, member of CG System Council, member board of trustee of ICARDA, ERI, Diversity International, and that indicates the profile of a person from national to the international level. Well-read person, and also creativity is there in him. That is reflected in the topic which he has chosen to deliver a lecture in this session. Very interesting lecture. Nature versus nurture. And that indicates his innovativeness. With his experience, with his role in different capacities, we are very keen to look forward for his word of wisdom through this lecture.
Very good afternoon to everybody. Dr. P.L. Gautam, chair of this session, Dr. Anjali, all the dignitaries of the dais, Professor R.B. Singh, Dr. Vassal, in fact, many others, Dr. Datta, Dr. Dilon, Dr. Joshi, Dr. Vakra, many others, in fact, Dr. A.K. Singh, all the delegates, participants in this Agricultural Science Congress. Initially, I was a reluctant, but subsequently, I agreed to come. And while again debating what I should be talking about, so I thought I should be talking something a little bit away from routine. Everybody talks about what all is happening all around. And with my own experience, putting bit of a policy perspective always is expected. But I thought there will be other people talking about that. And let me choose something which is different. But in the process, where time is limited, I don't want to really take more time in this introduction. But in the process, there will be a bit of science which might trouble some of you. So while choosing this, I consciously chose this. And I thought probably student and faculty members would also be here, being a big university. But that is probably is not that. So those who are not very familiar with the subject I am going to talk about, there could be little difficulty, but I'll try my best to explain. And I will only summarize rather than going to the details of what is actually included in those slides. And if I explain, that would also take more time. So I would like to give message rather than going to the data and the specifics. So nature versus nurture, new paradigms in the context of challenges to food and nutrition security. What it means, and as we go along, so you'll see this, because it is more rectangular in the projection area, it has traced in the bullocks. So the all slides will be also distorted like this, as we go along. So we have sustainable development goals before us. 2030 is not too far. Many people might have presented this. I don't want to really speak on this. But just to highlight, Zero Hunger Challenge. But food is related to all the 17 goals, in fact. So whether life on earth, life in water below the sea level, or climate or sustainable city, you talk about everything. In fact, agriculture and food system are all related. So they are all challenges. And we are far behind. We are far behind to reach somewhere nearby these targets and the goals which have been set. So certainly in this context of eliminating poverty, hunger, which is again associated with agriculture, giving the rural household engaged in agriculture. So if we talk of elimination of poverty, so everything relates to agriculture. How do we really address this in the context of understanding nature versus nurture? Zero Hunger Challenge, as I said, the whole world is suffering in terms of poverty, in terms of malnutrition. Data is there, I'm not going to read. I'm not going to really take you through very complex data sets. But certainly, so the overall score, if you see, Zero Hunger Challenge. So it's not very encouraging though, and some of the regions, some of the countries, number is mentioned there. So 36 countries to be very precise, where it's a very serious matter. So we need to bother, and you can see next slide will tell you Africa, Asia, where it matters more, and why all of us sitting here, why Agriculture Science Congress must bother. It's not just coming and listening to a lecture or not attending and chit-chatting here and there, but taking very seriously the messages which are being given through the lectures and deliberations, and subsequently putting those points into action. We keep having Agriculture Science Congresses and all that, Kochi and now here, Pantnagar and subsequently somewhere else. How much we are able to implement and move forward. Certainly we are moving forward, so that is implementation happening. So the challenges are there, whether it is wasting, whether it is stunting, whether it is malnutrition overall, and anemia and so on and so forth. So these challenges are there. And not only that, malnutrition and food deficiency challenge, but also the climate change and water, those issues are also there. And estimates are there. There are different estimates. Sometimes we question these indices, and sometimes we question these gradings which are done internationally. But even if we take the same set of parameters which are used to assess the situation in all countries, so it's a comparative. And if we compare, obviously we are lagging behind, we are far behind. And in this particular case, with regard to water, will be badly placed by 2040. So this remains a challenge. Per capita water availability reducing, and when it goes down to 1100 or 1200 cubic meters, so per capita, so obviously will be water stressed. That could be fights. And of course, the kind of negative impacts of climate change is there. Maybe somebody has already presented this slide. And what extent the crops are going to be impacted. This is our own assessment. You can see different crops, commodities, how they are going to be impacted, to what extent in years to come because of the rising temperature, carbon dioxide, and so on and so forth. In this context, this is very brief. So in this context, if we revisit the nature versus nurture debate, which is quite old, and for some of you maybe may not have heard about it. So I will very briefly explain, because time is always very limited. So William Shakespeare first mentioned about it in his monumental work, The Tempest. And this is the expression, a born devil upon whose nature nurture can never stick. So that was the kind of expression in The Tempest. So that was the first mention. And some literature states that it was even described before Christ. Some description is available. And it was John Locke who in fact described it in a different term. And he says probably in terms of human behavior and psychology, that at birth probably it's a blank slate, and through experiences, exposures and all that, through knowledge which we gain, so that will be writing on the slate. And subsequently we evolve as human beings, what we become in later stages. So human acquire all or almost all their behavioral traits from nurture, from the environment. So that's what another viewpoint in terms of a previous one. Nature was highlighted to be of paramount importance, and nurture doesn't really impact as much as it is described in Shakespeare's The Tempest. But here it is the other way, that it says that it's a nurture which saves everything and anything. And it is the inheritance of acquired characters. Obviously, it was not accepted, the Lamarckian view, but we are slowly coming around this in recent times. And this is the point which is going to be highlighted in subsequent slides. How the Lamarckian concept, which was again another extreme viewpoint at that point in time, in 1809, that how use and disuse determines subsequent evolutionary shape and directions. The neck of giraffe is another example which was often cited. And we didn't accept, and till recently, that probably evolution is not directional. The random changes happen, variation happens, and nature selects the best, and the best survives. The survival of the fittest. And Darwinian evolution theory operates. So that is how we have been moving forward and believing strongly until recently, which talked about probably there is a twist to this whole concept, that there is certainly a direction in which changes are happening. And the changes which are because of the environmental influence, they get transmitted, retained, transmitted. In the process, it becomes more directional rather than something random happening and getting selected. And this is a very important point in the context of nature versus nurture debate. And these two laws I'm not going to read, and hopefully you are looking at it, those who are interested. And so this is basically the philosophy. And in fact, Francis Galton is the man who gave many things, some of the aspects were a bit controversial at that point in time. And he certainly conducted some twin studies and took very scientific observations and talked about the influence of environment on the genes, their expressions, and how one becomes in terms of behavior, in terms of whatever our nature. So although he concluded that nature has a larger effect than nurture, so certainly he gave more emphasis and importance to the genes and inheritance of those traits. But he did mention and describe that nurture does influence the environment, does influence our development. And in 1903, the all plant breeders know this, Johansson's famous experiment that phenotype is genotype plus environment. And subsequently, of course, we brought in the interaction component. It's not simply stated as G plus E, but it could be a multiplier effect in many ways. Famous equations, very simply stated. And obviously, genotype and environment impacts have been known for a long time. And this was, again, Donald Hebb, 21st century, shows that both nature and nurture are very, very important. And he very emphatically stated that the question with regard to nature is important or nurture is important to one's personality, he very emphatically stated that which contributes more to the area of the triangle, whether it is length or width. So obviously both are important, and that's what it is. And that's what we have been also dealing with and treating, that agriculture cannot just be based on a genotype, a variety, but obviously environment is important, good agriculture practices are important, and that is how we have been taking it forward. In the context of climate change, in the context of the kind of challenges that we have all around, we need to really look at it critically and see that how it should be really dealt with. It is nature versus nature versus nurture, or nature and nurture together, or it is actually nature expressing via nurture. and which is evident from this particular paper published by Ridley in the year 2003. And an organism certainly utilizes both, and influence of environment cannot be undermined, and via nurture it expresses. And this is a slide which is a bit complicated, but I'll simplify it. We start with the genes, that is the nature. On the top, that is genome, sum total of genes carried in our chromosomes. 12 pairs of chromosomes in rice, 23 pairs in human, and it varies from species to species. Chromosome may be same in two different species, but gene content, their arrangement could be very different. The genome sequences adequately, plentifully reveal those peculiarities which are there in different genomes. So we start with that genome, the gene content, and go to the phenotype. The sum total of all the phenotypes is a phenome. And when we study each one of them, we have a battery of omics, genomics to phenomics, and in between all kinds of omics science. And we do omics and study each one of them in depth. But what is very, very important is environment, which is mentioned to the right of those and controls each one of them. Often it was believed that genome, the genes per se, are as such not influenced by environment. But today we say, and that is the topic of my presentation, that how environment influences genes and influences their expression by way of modification in the sequence, not in terms of the DNA sequence, but other modifications thereof that influence all those which are there, which is downstream of the genome. Whether it is transcriptome, whether it is proteome, the proteins, transcripts, or metabolites, all that, they are all influenced by this modification that happens at the level of genome. And those modifications are crucial, and they are influenced by the environment. And that's the reason why the context of climate change, they have become so important and now occupying this central place, this center space. And managing that is becoming very, very crucial, though often we neglect them. And probably this is time to really bother about it. And this is one example that how environment, extreme example, that how environment influences sex. And sex, in this particular case, bees, is based on what we feed, not genetically determined. Gene has per se don't determine sex, but what food is fed, that determines the sex. And particularly the royal jelly, who consumes and who is fed, and that determines who is going to be queen and who is going to be the worker. So this is an important example, and we know what happens. Many things are there. What happens gene transcription level and post-transcription level, and how gene expression influences all this. The food then influencing whole lot of things at the gene expression level. So I will take you through this story. It's a complex story, but I will only simplify it by giving statements, overall statements, so that the messaging is done. And here comes this branch, sub-branch of genetics, which is called epigenetics. I say it is a sub-branch of genetics because without gene, epigene doesn't exist. So epigenetics is there because of genes and genetics. So, and it is both cell-specific expression determines what we are today. The twins in two different environments develop differently. They might have different behavior. Identical plants having same genetic makeup can give you different expression in different environment. And we say genotype environment interaction. And we know there's a model, Leverett-Russell model, to understand that how genotype and environment interact. And if rank order remains same in two different environments for a variety, we say it is a stable variety and genotype. If the rank order changes, the genotype behaves differently in different environment, we say that it is an unstable genotype. And if we say that over generations a genotype behaves similarly, we say that is genetic plasticity. And why genetic plasticity? Genotype remains same. In different environments it remains constant in terms of behavior. In contrast to another which changes its pattern, its behavior. Why it happens? Because of subsequent expressions that happens in terms of genes and the proteins. So the genes remaining same, their expression differing, the rank order changes in different environment, and we say it's an unstable genotype, and we have to breed for stable genotype or genotype performing specifically good in a particular environment. Or when we talk of hybrids, the hybrid again a beautiful example of gene expression via epigenome modifications and wonderful expressions of genetics in terms of specific combining ability and general combining ability. My plant breeder friends who are sitting here, and they see how genes via expression influenced by a combination of genes which come from two different genotypes in the hybrid, influencing expression of each other and bring that kind of synergistic effect in the form of heterosis which expresses. As a beautiful example of epigenomics operating there. I will not be explaining everything in this lecture because time is limiting and all of you may not be interested. But it's a very, very fantastic in terms of science, exciting in terms of science and gene control mechanisms, gene expressions in different environments, and also within the cellular environment because of different things coming together, genes and genetic components and complexes coming together, and also in the form of genes which come from wild and unadapted germplasm to the adapted background and creating havoc at times and creating also changes in gene expression. And these are all the concept of epigenetics. And epigenetic patterns, they are serving as important bridge between genes and the phenotype, the genome and phenome, as you saw in the previous slide. And that is the link. And because all subsequent ones, the metabolome, the proteome, all those get subsequently modified accordingly. And there could be additional changes in protein because of environment, which is not even related to the gene and the transcript. And that is again another layer of complexity which is brought about, and epigenetic alterations which are brought about. Very few things I'll describe before you to simplify these cases. I will not really read and bother you, every word of it. And this is actually Conrad Waddington, who coined this term epigenetics in the year 1942, and to explain development, how development is happening, because the cells and genes remain same, but there is a hard tissue. that is a lungs tissue, that is a kidney tissue. So different organs are being formed from the same initial zygote. How it happens? In previous case I said sex is also being determined. Genetically they being same, there is no other genetic mechanism to determine sex, and it's being determined subsequently. And in this case, how different organs are being formed? Initial zygotic genetic content being same. So this is a very interesting one, and that's time the development was explained with the context in the context of epigenetics. It says that epi is the Greek prefix, and which says upon, above, or in addition to, or near to genes, or whatever is contained in the chromosomes. So it's a beyond genes or above genes. So something beyond genes or above genes. So what is that? And this is what forms the content of my lecture today. And I'll tell you how interesting it is, it has become in recent times, and how challenging it is to deal with as we go along. And if you see, the epigenetic changes are mentioned here. If you see the left hand side, there are 4 types of histone. Here, what is given is H2A, H2B, H3 and H4. There are 4 types, and you have another type called H1. I'll not really talk about that. And this 2 of these 4, they form a structure called nucleosome, and surrounding this is DNA. And they remain together in chromosome. And if they are relaxed, gene expression happens. The relaxation of those requires acetylation. And acetylation of lysine component in protein, because histones are proteins. In that there is the amino acid lysine. And lysine, if it is acetylated by acetylase enzyme, so then you have acetylated histone, and that is called histone modification, one form of histone modification. There could be other forms of histone modification which are written there: methylation, acetylation, phosphorylation, ubiquitination and sumylation. So all these changes can happen to histone. Acetylation can influence gene expression. A gene being there doesn't matter. Sequence may not change, may not differ between 2 organisms, 2 individuals. If these changes are brought about by environment or by your food, then there will be a difference in gene expression. If there is a gene expression, then there will be a differential performance. There will be a differential impact of environment. So the climate change impact could be different. And that is why I explained a bit that why these epigenetic changes in the form of DNA methylation or histone modification like histone acetylation are very, very important and crucial. They may be heritable. They may go through the gametic pattern and get inherited through sexual cycles. These patterns, and they are called epimutations if they get inherited. And that is a bit scary. Because what you get through your food, a change that you get through your food can go to your progeny, your children. And that is a bit scary at times if you are not careful. So, they are called epimutations. And they are often reversible. That is fortunately so. So anyway, so that's the reversible pattern is indicated by arrows in both directions, while where acetylation is written. Lysine when it is getting acetylated. And this is one beautiful example of a epimutation in Arabidopsis thaliana. In Arabidopsis thaliana, because of this methylation change in DNA, in a gene called Superman,
So you will have more of anthers. So, and you can see in this picture, so you'll have more of anthers. When DNA sequencing was done, the mutant and the wild type, they didn't differ in DNA sequence. What differed was in terms of this methylation, and that is what is indicated there. And those alleles are called epialleles because they do not have DNA sequence alteration. So in that sense, it is not a genetic alteration, but because it is epi, it is added, it is above something addition. So this is a methyl group added there, and that's why it is called epiallele. It is transferred through sexual cycles, though there is a certain degree of instability, and that was published in Nature, in Science. So that was it's down below. Yes, it is coming. Science, and also you have another published in Nature. It's again a very interesting story in Linaria, where the orientation of flowers is altered and again inherited because of epimutation, not a sequence change. So there are so many examples now of epimutation. Earlier we did not know this. We had absolutely no idea that such changes are there, and they are agriculturally so important, not just in plants, but also in animals, but also in feces, but also in other organisms, in insects. So they are so important. So the epimutation, a change, the epigenetic change, giving epialleles and impacting trait expression, getting inherited through generations, and there are examples, and this is time for us to bother about it, more so in the context of climate change. Okay, this is another example how flower orientation is changed. So this is a beautiful example in case of rice, a recent example. And this epimutation gives semi-dwarfing, rather dwarf phenotype. Also, you can see the number of anthers there. That's also a transformation. It's the abnormality which is created. It's not a desirable change, though, but it happens in nature. So it is undesirable epimutation. Instability is also seen, and two traits, the plant height and the reproductive organs, getting influenced. And you can see what is highlighted is the methylation, the vertical bars in the top, the left hand side, if you see, the vertical bars indicate heavy methylation. Lack of vertical bars shows that there is less of methylation. So less of methylation means gene expression. Heavy methylation means there is no gene expression happening for this particular gene, FIE1. And if there is gene expression, then what happens? The corresponding phenotype you have seen. And in evolution, to have normal plant phenotype, methylation is required. In domestication process, during selection, we have selected for more methylation. So methylation sometimes is desirable because it is gain of trait for us. Agriculturally important trait can be gained by way of methylation. Loss of function can also happen by way of methylation. So both ways are important. So we need to really understand this. And this has become so important in the context of agriculture and human health. This is another example that how the ESP gene in case of rice. Most examples you will find from rice because that's my interest and my work is on also rice, recent work. And you can see the panicle size. The panicle size that we have selected today in the cultivated forms and the kind of panicle, the mutated format, the epi-alleles which are indicated there towards the right side. So you can see that those are undesirable kind of changes. Again, because of epi mutation. And this mutation has happened, epigenetic modification has happened in the ESP gene. It happened in a non-coding, long non-coding RNA. Non-coding RNA. It's a long non-coding RNA, and that non-coding RNA, if it expresses, it influences expression of other genes that impact these genes. And how this has happened in evolution, you can see through the domestication process, we have created all these changes. And these changes without realizing what we have done. When we say domestication, what is domestication? Domestication means altering gene expression. And right kind of modifications are selected. And these modifications are through these epigenetic means. And beautiful examples are there. And these are few examples which I have highlighted here. And you can see the upper one, the wild type, and how methylation happens, and the lower one, the demethylated ones, the long non-coding RNA expressing. Once it expresses, there will be suppression some of the useful genes. In the process, you will have undesirable panicle phenotype. So these are all epigenetic alterations. And not only that, it has happened in the process of domestication. And what is adaptation? Adaptation is all about epigenetic alterations. And how we carry out this by repeatedly growing organism, cultivating them. There is another session, I believe, after this. I don't want to really delay the next session. And this is a beautiful example of OSSPL14. And now this gene is being used in marker assisted breeding. Dr. Sundaram is here, and they have already done this work. ED has done this work, and this is actually a PLE. This is actually a PLE. There is no sequence difference. It is a PLE, and that gives plenty of grains per panicle. So panicle becomes heavy, the heavy panicle, and that is being targeted in marker assisted breeding. Means if we can discover more and more of these AP alleles, and we can target improvement in crop plants, we can have domestication, we can have greater adaptation, wider adaptation, if we can discover, if we can manipulate the epigenome of crops and animals and fish. And that's what is the message I would like to give through this slide. Not only that, the yield, the plant type, grain shape and size, all these are influenced. Even the quality, the nutritional quality also gets influenced. And this is the example, beautiful example in case of tomato. Naturally occurring APLE that determines vitamin E content or accumulation in tomato fruits. Now, another example of epigenetic mutation or APLE, epigenetic alteration that influences the nutritional profile, and we need to really understand in the context of greater adaptation, in the context of nutrition, nutrient accumulation, and in the context of climate change, all these epigenetic mutations and alterations which have been accumulated over years. Let me go fast and finish it. And this concept of environmental epigenetics has come to the center stage today because of climate change. And this journal, Environmental Epigenetics, has now come into existence. And this has special issues on epigenetic alterations which are influenced by environmental changes, whether it is drought, whether it is salinity, whether it is heat, whether it is submergence, and all that, and how they influence gene expression, and how important it is to manage those alterations by different means. And this journal describes that special volumes, and you can go through this, that climate change impacts on all organisms are mediated through environmental epigenetics. to influence genome activity, to promote the physiological change and phenotypic variation for the organisms. Since this is not only involved in the direct exposure impacts, but can be inherited, must underline this, but can be inherited to subsequent generations through epigenetic inheritance. The short-term and long-term impacts of climate change involve environmental epigenetics. And I believe this is the time that we must all worry about and work on this very intensively to understand what is really happening and how far we will be able to understand and manipulate it and address those in certain terms. We have to really understand. We don't understand at this point in time. Globally, the research is actually beginning to understand what is really happening. And this is taken from a review that if there is stress, whether it is drought, salinity, heat, and all that, there could be epigenetic alteration, which is indicated there. That will be APLL1, APLL2, APLL3. APLL1 could be a short-term adaptive measure for the same generation. APLL2 may get inherited. APLL3 may get inherited. APLL3 may get inherited in subsequent generation and in the process get fixed in the form of AP mutation. So the environmental factors may create AP mutation, and AP mutation can be actually carried forward, and for 20 generations they have been found to be actually taken forward. And right hand side, which is indicated there, if you cross two individuals, particularly in case of wild crosses, there could be AP alleles which are created and get fixed, giving different traits. There are all examples in Arabidopsis thallium. It's not just mutation, reshuffling of genes. It's not just reshuffling of genes, mind you. It is epigenetic alterations which happen when two genomes are coming together. And that can bring you different traits which are expressing in progeny generation, which often call it the transgressive variation. Transgressive variation might happen. It could be altogether new variation. It might happen because of this epigenetic alteration that happens because of interactive effect of two genomes which are brought together by way of crossing. And this is what in Arabidopsis example is there. And this is what is really important, that these changes we studied in rice, we said how it is really carried forward. We saw that a significant, about 4.5% increase we observed in the overall 5-methyl cytosine content in a drought-tolerant variety Nagina 22. Nagina 22 is a rice variety, which is a house-type variety, which is an upland variety, which is drought-tolerant, which has been sequenced and elaborately studied. And when we expose this to drought versus IR64, we find it contains more of overall 5-methyl cytosine is more in drought-tolerant one. And more importantly, what is important here is to know, this is a recent paper of ours, 25% of the increased methylation level was retained even after the withdrawal of a stress. Mind you, stress tolerance is because of, say, methylation or demethylation, whatever happens in drought-tolerant variety. Some of those genes which are responsible for susceptibility, and they are actually silenced. And is there a time limit? So you can let me know how much time is there. I can go fast. Oh, okay, 5 minutes. So I'll finish it up. So this is actually taken forward, 25% of these changes, and to subsequent generations. So this is very, very important. And this is another paper which highlights another important point, that Nagina 22 versus IR64. But more importantly, if you grow on DSR condition, direct seeded condition versus transplanted condition, what happens in Nagina 22? Not all variety will perform well under DSR condition, and there is epigenetic alteration which is controlling that performance. And this is a clear evidence in recent paper, 2024 paper we have highlighted that all varieties do not respond equally to DSR because of these epigenetic changes which are happening there. And Laguna 22 is better suited than AIR 64, which is evident here in terms of performance, root architecture, in terms of other attributes, and also gene expression, which is highlighted here. And there are other papers which very clearly, and this is one which is inherited for generations, and you can see 11 successive generations. 11 successive generations, this change which happened, this epigenetic alteration which happened, carried forward for even 11 generations, and that is an adaptive epi mutation getting created and carried forward, and we have to really bother about it, work on this. And not only that, food exposure also matters. Food exposure for plants, what we are feeding. In this context, good agriculture practices, rather best agriculture practices, I would say, that matters. We often don't bother, and the natural farming system we talk about, and the regenerative agriculture we talk about, and all that matter. Food for plants is important. What gene expression will take place, even if their genes are sitting there, they may not really good to give you performance. We know this, but through epigenetic means we know very well, it's very well established. And also as a human, what we eat, as animals, what we feed to them, it also matters. This is very, very important, and I highlighted this in the National Genetics Congress, which happened recently. And again, I'll highlight today that this is called nutritional epigenetics. There are so many food items we eat. We say food doesn't matter, but food matters. Why? The microRNAs that we eat, they influence our gene expression. The microRNA that we eat, they are non-coding RNA. The RNA that we eat, they might influence our gene expression. And that gene expression might influence many diseases. And this is what is highlighted in this particular paper. And if you are interested, you can read this. And also, it has shown that it passes through the system, it gets absorbed. It has been observed. And this paper you can read if you are interested. It passes through and absorbed, goes to the cell, and gene expression is altered. So, in different tissues, heart tissue, lungs tissue, in different tissues. So, I am not frightening you, but this is the fact. And then, there are specific examples that how different microRNAs or food items, they influence the epigenetic function of the genes. And you can see anacardic acid, curcumin, diallyl sulfide, caffeic acid, quercetin, and so on and so forth, how they influence the expression of our genes, okay, by way of modifications. Histone, acetylation is important, I highlighted. And that acetylation process is influenced by your anacardic acid. Histone modification, if histone is modified, gene expression is altered. And if that happens, we will have a particular phenotype, a particular function. And we have plenty of these adaptive changes in our local germplasm. We must conserve them because evolution is happening in nature. Evolution doesn't happen in gene banks. We have arrested in gene banks. We have arrested evolution in gene banks. Evolution happens in nature. Adaptive changes happen in nature. They accumulate. They can get transmitted from generation to generation. They can give us climate resilience. They can give us good nutritive value. But they have to be maintained there. Who will maintain? Farmers are maintaining. We must support them. And all of us must go back to the field. On-farm conservation has to happen. On-farm cultivation has to happen. Germplasm should be taken there to the field so that they evolve and they give us these adaptive signatures for all of us to study. And AP genomics we do, pan genomics we do. Now it's the time of AP Pangenomics. If Rajiv Varshney is here, he will be here. He will carry forward this AP Pangenomics. You don't find globally papers on this. And we must understand what is really happening to the epigenome at pangenomic level. Epigenetic changes which are carried forward by our traditional varieties in plants, in animals, in fish, so that they are there for future. And this is what is actually, and you can manipulate. There are ways and means to manipulate the epigenome. The epialleles can be created for future. And I will not go into details of it. It's a very complex slide, but this is the message. You can manipulate very specifically in a directed fashion and go ahead. And what is the message? Nature and nurture both are relevant and important, and we must mind both. We must mind both. Nature and nurture are important, and we must mind both. More intensive studies of epigenetics, epigenomics, pan-epigenomics, otherwise you will not understand what is there in the adaptive signatures in the unadapted germplasm. And also epitranscriptomics. I didn't highlight. We have paper on epitranscriptomics. Now, what is really happening at the transcript level and how stability is influenced. Stable epialleles discovery and use is very, very important for animal improvement. For fish improvement, we have fish with 50% more yield, but we must understand what is those adaptive change. And we must understand gaps and I would say BAPS, best agriculture practice for and with balanced nutrition. MicroRNA component of food and feed system. Feed system. What kind of feed we are giving to the animals or fish and what way they are influencing gene expression, that matters. We must be bothering about it. We must study this. And this is the message I would like to give to you. So science and everyday life cannot be and should not be separated. Science for us should be a partial explanation of life and complete explanation lies elsewhere. So there is more to learn from nature. We are yet to understand nature. And this was the statement of Rosalind and Franklin. And we must really go by what she stated. And thank you, Farmer, if you have it today. And thank you very much for your kind attention. Thank you, Chair, for giving me time for taking you through what I wanted to give you. So, with this, I stop here. Thank you very much.